of the day, Hon'ble Nobel Laureate Professor JeanMarie Lehn, Honorable Professor Dr. R.S. Bawa, Pro-Chancellor Chandigarh University, Honorable Professor Dr. B.S. Sohi, Pro-Vice Chancellor Chandigarh University of the day, respective Executive Director, Nobel Laureate Directors, JeanMarie Lehn, Principals, Honorable Professor Dr. R.S. Bawa, dear students. Pro-Chancellor, on behalf of Chandigarh University, I, Dr. Renu Sharma, extend a very warm and heartiest welcome to each one of you present here. Today we have a very eminent personality, Honorable Nobel Laureate, Professor JeanMarie Lehn with us to share his views on steps towards complex matter in chemistry. Professor JeanMarie Lehn is a French chemist. He received the Nobel Prize in Chemistry in 1987 for his synthesis of cryptons. So, without much ado, I would like to invite Honorable Pro-Chancellor, Chandigarh University, Professor R.S. Bawa, to welcome Professor JeanMarie Lehn. Sir.
Thank you, Professor Renu. Honorable Professor JeanMarie Lehn, the Honorable Nobel Laureate and a French scientist, chemist by profession. Sir, we are delighted to extend a very, very warm welcome on behalf of our Honorable Chancellor and the whole Chandigarh University family, which consists of more than 6,000 members, 60,000 members, including 30,000 students. Sir, we not only feel honored, but we feel greatly excited because I'm sure that all of our students and faculty will get a great source of motivation and inspiration through your talk today and your motivation today. My dear students and faculty members, I feel again greatly honored to share a small introduction about the great Professor JeanMarie Lehn, who is presently working as director at Laboratories de Chimie supramolecular ISIS University, Louis Pasteur, France, Paris, and who is also director at Nanotechnologies Institute of Research, Center of Karlsruhe since 1998. Coming from a small place in France, he had his undergraduate studies in Strasbourg and then graduated from University of Strasbourg and completed his doctorate in 1963 from University of Strasbourg. My dear colleagues, as we say that coming events cast their shadows before, right from 63, the Honorable Nobel Laureate started getting awards of different nature. And he has done his postdoctoral research fellow at Harvard University and worked on vitamin B12 total synthesis with, again, great Professor Robert B. Woodward. And he has worked in 14 prominent universities of the world and worked in esteemed positions. The Honorable Nobel Laureate got 13 prizes and awards before he got Nobel Prize in 87. So, sir, this is a unique journey, a great journey, and I think a great motivation for everyone who listens. Before getting a Nobel Prize in 1987 in chemistry, he got 13 prizes of different type. I'll just mention a few of them. Raymond Bird Prize for Société Chimique de France, Gold Medal for Academic Pontifice des Sciences, Gold Medal of CRRS in 1981, then Pierre Brouillantes Medal, and many more medals before he got Nobel Prize, and 13 medals before Nobel Prize, and 22 medals after that. And again, I'm sure all of us will feel greatly honored to hear that he has in all 36 awards and medals. He is membership of 50 prestigious professional societies across the world. And these awards have been given by more than 10 countries and many universities. More than 790 publications to his credit. And the work in areas of theoretical organic chemistry, dynamic nuclear magnetic resonance, supramolecular chemistry, cryptates, anion coordination chemistry, bioorganic chemistry and biological applications, dynamic combinatorial chemistry, and many more. In fact, all these are modern areas of chemistry. And sir, without taking much time, I once again extend a very, very warm welcome, and we shall remain beholden to you for all times. And a very, very warm welcome to all those who have joined my own faculty, the guests, and I am sure thousands of people who are on Facebook and other social media who will be delighted to listen to the Honorable Nobel Laureate. Thank you so much.
Thank you, sir. Now I request guest of the day, Professor JeanMarie Lehn, to enlighten us with his wisdom. Dear students and faculty, you can write your queries in the chat box, and at the end of session, these will be taken up by Professor Lehn. Over to you, sir.
So, my turn, yes? First of all, thank you for the invitation. It's a pleasure to be, even if it's by remote means, in Punjab at your university. I certainly would have preferred to be there in person, but okay, we can do it this way and we can meet, or at least we can see each other if we don't meet person to person. Thanks for the invitation. A new university, I've never been there, so a new university is always something which is very interesting for me because universities represent the knowledge of tomorrow and of the researchers of tomorrow and those who will later on lead mankind on better ways. So my lecture is a very general lecture because I understand that the audience comes from different origins, from different expertise in science and technology. That is why I have chosen to make it a very general public science lecture. And in fact, the title is Steps Towards Complex Matter. And I would like to show how crucial role chemistry is playing in these steps towards complex matter. Now, what are these steps? We have to start very, very early. We have to start 13.7 billion years ago. That's the start. Our universe was born with a Big Bang. After the Big Bang, there was no chemistry. It was much too hot. There were no chemical elements formed. But then the universe cooled rather quickly. And this first period is the period of physics, no chemistry. But when it cooled down enough, then the particles which had been formed led to the building up of atoms, and atoms could get together to form molecules. And that is about at this time where chemistry started, the age of chemistry, about after 300,000 years after the Big Bang. Then molecules became more and more complicated by processes we don't much many of, but we get to understand them better and better. And they made molecules, bigger and bigger molecules. Molecules began to aggregate, to make compartments. And at some I did it several times already. I split the screen. We will be back on. Do you see it?
Yeah.
Okay, then let me put it full screen.
Full screen.
Now you see it, yeah?
Yeah, right. We can see it.
Yeah. Okay, so this is finished. So I was at a point where I said that chemistry then developed and made molecules, more and more complex ones, and at some stage a new property appeared, which was life on our planet Earth. And that's the age of biology. But it did not stop at this point. It continued, and it turned out that a new property, even more important, appeared, which is thought, thinking. And I show you here this statue, this from a French sculptor, Auguste Rodin, which is called the thinker. This is the thinker. And these are the steps towards more and more complex forms of matter. And thinking matter is the more complex form. What I mean by that is, first of all, French, what is matter? Now, this looks like a dark screen. It is a dark screen. This is because, and these are the steps nowadays know that there is 68% of dark energy in our universe, 27% of dark matter, which means 95% darkness. But there's 5% visible matter, and this is the matter that matters. We are part of this 5% of visible matter. So the evolution of matter therefore started with divided matter, particles. It became condensed matter, molecules and so on, and molecular aggregates. Then it became organized, living, thinking, and even some more complicated form of matter, which we cannot know about for the moment. But thinking matter is for us the highest level of complex matter in our universe. And this is, so to say, the evolution through different steps towards more and more complex matter. Now, what is the big question then we have to ask? The very big question, the most important one probably, question is, how does matter become complex? How did matter lead to a thinking organism like us, like human beings? And how did these elementary particles, the initial ones, lead? By which way did it lead to thinking organisms? And maybe there are even higher forms of complex matter. So the answer to this question is a very fundamental question. That is the reason why we exist, but how did it happen? And the answer is provided by science. Mankind did a fantastic job in inventing what we call science. Now, science, if we consider the three main pillars, is, of course, first of all, physics. Physics deals with the general laws of the universe, the way everything functions. Biology deals with the rules of life. How does it operate? These are not laws. The laws are built up, studied by physics. But depending on these laws, life has appeared and life has certain rules. So what is chemistry doing? Chemistry has a very, very important job. It is to build the bridge towards complex matter, to build the bridge from the general laws of the universe to their expression in a specific way, in specific entities, living organisms, thinking organisms. One may even say that the structure of our universe is such that this property appeared spontaneously by self-organization, because the structure of our universe is such that it will self-organize. And one even can consider that as a cosmic imperative. I mean by this also that, if I come back to my early first slide, that at the end where we are now, 13.7 billion years after the Big Bang, the evolution will continue, self-organization will continue in some regions of the universe, and life is present very, very probably in many places, probably also thinking organisms. We are not alone. We are just a point in space and in time. But a point, this entity called human beings, and this feature, this property called thinking, led us to the discovery of the structure of our universe, the intimate structure, which is something absolutely fantastic to be able to say, we human beings coming out of the evolution of the universe, being just a point in space and time, nevertheless, by the power of thought, of science, are able now to understand, at least to a large extent, but of course there's still lot to discover, but to understand by a large extent what our universe is made of and what we are made of. So this self-organization I'm talking about has two aspects for the moment. First of all, there's the cosmic self-organization, which is the large-scale self-organization through gravitational forces, the Einstein forces. And this leads to the formation of the cosmic structure, where the gravitational forces operate on the initial homogeneities in density and in rates of expansion at very early times after the Big Bang. But this is not for us. This is not what sort of concerns us intimately. It is very important, of course, but what concerns us directly is the self-organization of complex matter, which is through electromagnetic forces, which act on the bricks of matter, which act on the bricks which have been generated in the evolution of the universe. And this is then the formation of complex aggregates of elementary bricks, which led to organized living and thinking matter. So the chemical evolution of matter is, has to do with starting at the prebiotic stage, before life existed. As I said, the universe generated progressively more and more complex molecules, which then evolved. But this evolution had anything to do with biological evolution, with Darwinian evolution. There was an evolution of matter towards more and more complexity by self-organization of non-living matter before life existed, which means that evolution is a much broader concept than Darwinian evolution. It existed much before life. So biological Darwinian evolution is in fact also a chemical evolution, because it deals with these changes, the evolution of entities, living organisms, which are built on chemistry. All the living organisms are, of course, composed of chemical entities. So chemistry is usually said to be the science of the structure and transformation of matter, non-living matter and living matter. We will see this is what chemistry has to do, how is it built, and how can you transform matter? Pieces, that means entities, molecules into one another. There is another property which will come up later, and which is a very important one too. So for a long time, then scientists investigated the structure of matter. They found that there were bricks, that there were things there which defined the objects of matter around us. But it was not very clear how this was organized until the middle of the 19th century, when several chemists, but one specifically, made a big step, which is that Dmitry Mendeleev formulated the organization of the bricks of the universe, of the bricks of visible matter. In 1869, Dmitry Mendeleev in Russia, then he published a very important paper. I show you here really the original publication. This is a picture of the original publication starting here. which had to do with the relationships between the properties and the weight of atoms, of the elements, on the relationships between the properties and the atomic weight of the elements. Mendeleev in 1869 in a German journal. It was written in German. And what you see here on this page, this first page of the paper, is an organization of the bricks of matter, the elements which were known at that stage. And they are, you can easily, for the chemists, they easily recognize, for instance, here boron, carbon, nitrogen, oxygen, fluorine. There is also carbon, silicon, and there is a question mark. Interesting. What is this question mark? Mendeleev was convinced that his table was correct, and when there was something lacking, he put a question mark and he said that we will discover this, for the moment, unknown element. And of course, they were all discovered. So this was extremely powerful to be able to organize the pieces which make up visible matter. This is now called the periodic table of the elements. And as I understand, I show you here, it was the 150th anniversary 2 years now, in 2019 was the 150th anniversary of that. Now, nowadays, this periodic table of elements is this. This is what it is presently. Here, what you see is a full table starting with hydrogen here, and in natural elements, closing up with uranium here, 92 here. And what is absolutely fantastic here is that this table of the elements which make up the bricks of visible matter is a complete table. Try to think. This table is complete. These elements are present anywhere in the universe. If you go to a planet billions of light years away, they will be the same. Think about what that means. That means you are sitting in your chair. You are looking at this picture, and this picture tells you that if someday I meet somebody who I can go to a very, 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 very distant place in the universe, these elements will be the same. I find that when each time I look at this table, I say this is incredible that human beings sitting on a small, small, small place in the universe have used the power of inquiry and of science and of thought to develop this table to show that this is what the universe, the visible matter, I must insist again, is made of. Now, why do I say it is complete? Because it is like counting 1, 2, 3, 4, 5, 6, 7, and so on. Because hydrogen is made of 1 electron, 1 proton, helium, 2 electrons, 2 protons, lithium, and so on, and so on, and so on. So you can just count, and between 1 and 2 and 3 and 4 and 5 and 6, there's nothing between. So the table is complete. I must say it bothers me a little, because scientists should not be dogmatic. They cannot say that is it. But here I cannot see any way which would tell us something else between these elements. These elements are present everywhere. And when astronomers look, or astrophysicists look at very distant places in the universe, the elements are the same. They are the same. And we know them. This is incredible. And this is the pleasure of chemistry, the playground of chemistry. Chemists are like children. They play with these building blocks. They assemble them following, of course, given rules, which are the rules which physics imposes onto them. And they play around and they make all these combinations possible. And we are part of that combination. And we as human beings are a specific, extremely complex combination of these elements. That's what we are. We are a lot of carbon, a lot of hydrogen, a lot of oxygen, a lot of nitrogen. That's what human beings are. But they have a brain, and that is the important thing. So from the atom to the molecule, molecular chemistry developed, and developed in a way which chemists call linking atoms by strong bonds, which they call covalent bonds. Strong links between the atoms make up the architectures called molecules. And we can look then at some milestones in molecular chemistry. Let's look at two milestones just to give it an idea. In 1828, a German chemist, Friedrich Wöhler, made urea, the molecule you see here. That's a very simple molecule. Very simple. It has one carbon, one oxygen, two nitrogens, and four hydrogens. Very simple. But he did this in the laboratory. He made urea in the laboratory, starting with a compound which was a different chemical compound called ammonium cyanate. Now, the important thing was there are two things which are important. Number one, Wöhler was able to transform ammonium cyanate into urea. That's a chemical transformation. Secondly, by doing so, he showed that between the molecules present in living organisms, urea is in urine, and the molecules present in not present in living organisms, like ammonium cyanate, there is no abyss, there is no big let's say, split between the two. It's the same world. It's the chemical world. The elements of the non-living matter and the elements of living matter, the molecules of non-living matter and the molecules of living matter are just the same world. And that was very important because at that time, philosophers thought, and scientists thought, that you could not make something contained in a living organism from entities of non-living type without the help of what they called a vital force. And the synthesis of urea from ammonium cyanide destroyed that and showed it was a myth. That's the same world. Now, 150 years later or so, this very complex molecule, vitamin B12, was built up by what is called total synthesis. That means building it up from the ground by putting together carbons, hydrogen, oxygens, nitrogens, and so on. This was a fantastic effort. It was considered at that time in the 1970s as the, so to say, the Everest of chemistry. And it was done by the common efforts, the combined efforts of two groups, one at Harvard in the USA, in Massachusetts, and the group was directed by Robert Burns Woodward, and the second one, working together, both were working together, by the group of Albert Eschenmoser at the Polytechnical University in Zurich, in Switzerland. I was at that time a postdoc with my one-year postdoctoral stay with Woodward in 1963-64, just for, yeah. And so this is, of course, a fantastically complex molecule compared to urea, and it showed that molecular chemistry had become very powerful. Of course, it continued, it still developed. Nowadays, one would make vitamin B12 in different ways, probably more efficient, better yields, and all that and all that. And many new reactions were discovered, new processes, new compounds were made, new drugs, new materials, and all that developed. And it will continue to develop because molecular chemistry is still, and of course, it will remain a very active and fundamental science. But we have then to ask a question. What else should we consider in addition to looking at molecules as such? Let me illustrate that in the following way. Here is a blue sphere, which is a cancer cell, and here are two killer cells. The killer cells are supposed to find out the cancer cell and to destroy it. How do they know that the other one is a cancer cell? How do these partners know each other, that the killer cells can do that? Similarly, here is a white blood cell, artificially colored, And the blue dots are the HIV virus. When the HIV virus hits a white blood cell, it can infect it. How does the virus get the information, the fact that it has reached its goal? Something must happen between these two bodies, these two type of cells, and the cancer cell and the killer cells, and the white blood cell and the virus, which tells the partners that they have reached their goal. Of course, they shouldn't make the mistake. If the killer cells make a mistake, you have a big problem. So they shouldn't make a mistake, or something tells these entities that they have reached the goal. That means that there is, on the basis of molecular chemistry, another chemistry which we call supramolecular, which deals with the assemblies, which things getting together, which looks not just at what are molecules, but what happens when molecules get together. What happens in populations of molecules? What happens in assemblies of molecules? I call that a chemistry beyond the molecule, supramolecular chemistry, and this is relying on the interactions between molecules by other types of interactions than the ones which link atoms in molecules together. Three main properties have been studied over the years in this area of supramolecular chemistry. Molecular recognition, how do molecules recognize one another? Reactivity, how can they react with one another, or how can they carry themselves through barriers like membranes in cells. The most basic property is molecular recognition. Now, how can we express molecular recognition? We can do it in the following way. Molecular recognition first requires interactions. Things have to bind together. If they don't bind, they ignore each other. But that's not enough. You need information for recognizing. You cannot recognize without information. And this can be considered as being a sort of a, if I do it very, very simply, a sort of a double complementarity between the geometry of the objects, the way they look, their architecture, and the types of interactions which link these molecules together. There was a very early picture given of that, very early, but still a strong image of it, which is that proposed by Emil Fischer in 1894, when he said that molecules, in order to interact on each other, have to fit together like Schloss und Schlüssel, like a lock and a key fit together. This was in a very famous paper by Emil Fischer in 1894. Let me also say, because we are so proud of it, that Emil Fischer got his doctorate in Strasbourg in 1874. When he wrote this famous paper, he was in another place. He was in Munich, but I think it was in Munich. And so in 1874, he got his doctorate. Anyway, this image of a lock and a key is a very strong one. We now know that's a bit more complicated. The lock and the key are somewhat soft, they can adjust somewhat, but basically it's a good image, and it is the foundation of biological molecular recognition. Now, the most important information system built on recognition is the following one. It is the information contained in the genome of living organisms. On the right-hand side, you see the famous double helix, which is built on two strands of molecules, two molecular strands wrapping around each other, as you see here. So this is built up of a long string of connected entities here. You see this long string here, into which are fitted different letters. They are bound to it, and these letters have a given sequence. It is the sequence of these letters which makes the information in our genetic, in our genome, in our genetic information. Adenine, guanine, these were just sort of names given by chemists. Adenine, guanine, thymine, cytosine are very simple chemical groups. This is adenine, this is guanine, that is thymine, that is cytosine, very simple chemical groups. And it is these four letters which write the genetic information of living organisms. Now, this is the storage of information in this long string of a sequence of letters. It's different, of course, for each organism. But how can you read this information? The reading proceeds by contact. And here, the contact is expressed by these points. Here you can see. And adenine pairs up with thymine or uracil in this way by 2 points of touching, of attachment. And guanine pairs up with cytosine by 3 points. So it is a very simple binary system where the reading of the molecular information is done by 2 or 3 points of interaction, 2 or 3 points of interaction. And this is a binary system. It's very simple. So it's quite astonishing that living organisms, what distinguishes a mouse from an elephant or a tomato from a human being, is stored as information in this very long strand of letters and is read out and processed by this binary 2-3-2-3-2-3 process. So chemistry must be considered as an information science. All this is information. Information is stored through chemical letters. It is read out through chemical interactions. So the molecular storage is the way the molecules are constructed, and the processing of this information, which is present in molecules, is by interaction, by fitting together. So chemistry is the science of what you may call informed matter. And of course, biology is obviously one of the highest expression, is the highest expression of this informed matter, because an organism does not function without molecular recognition. In your body, as you are sitting there, all the molecules, everything that is happening in your body starts with molecular recognition. It involves molecular recognition. Otherwise, nothing works. So let me just very simply show here a picture where we have keys on the left-hand side, 3 different keys. We have a lock in the middle, which you can call a receptor. And of course, the red key fits in. So this question of trying to understand how molecular recognition functions has been studied by many, many groups over the years, very many studies in numerous laboratories of molecular recognition processes in India, also in many, many places. Many laboratories have joined the effort of understanding how molecular recognition occurs, how it operates, and of making artificial types of molecular recognition. So let me just also show you some applications. Of course, basic research is the way to gather the knowledge, and applied research is applying that knowledge. Applications in life science, the most important one is obviously drug discovery, because a drug is a molecule which is designed so as to fit with a biological target. A drug is a key for the biological lock, and we want this key to be as selective as possible so that it only binds to the target and it doesn't bind off target. So this is the most important application and the most important consequence of the studies of molecular recognition. Of course, people had discovered drug before that, but nowadays one has to understand how this happens, how drugs operate, and it, of course, it starts with binding to the target. We also have then developed, in some other cases, another type of systems which also relies on supramolecular chemistry. For instance, I show you here a molecule here which absorbs violet and UV light, and then transfers the energy absorbed to an entity, a europium ion. Europium is part of the periodic table of Mendeleev, and which then emits red light. And this has then been used to develop systems for medical analysis, immunoanalysis system developed at a small company by Dr. JeanMarie. Then a very important other feature is that I said it's recognition, reactivity, and transport. Transport, let me just say a few words about that. Transport is a process by which some molecules help others to go into a cell, especially. And gene transfer is one of the most important, if not the most important one. Of course, you can also do other types of transports, but let's look at gene transfer. Gene transfer is a transport process by which a piece of DNA, as small as a gene, which is a piece of DNA, is surrounded by a synthetic vector, that means by chemical compounds, which then envelope the piece of DNA and help it to cross the cell membrane so as to be expressed, translated into RNA in the cell, which then leads to the proteins, the product. So gene transfer is a way to make a cell produce the protein, the compound, which is coded by the piece of DNA, by the gene. And this is of great importance for biotechnology as well as for gene therapy. Gene therapy to correct for deficient genes in human organisms and in others too, but mostly in human organisms. However, I would also like to insist on one thing. The use of gene transfer and the modification of genes for biotechnology purposes, for artificial purposes, and this is called making genetically modified organisms. I consider this as one very important aspect of the question, because this is a way for us to modify organisms and to make them do what we want them to do. Now, people are afraid for very silly reasons, and many media are propagating very bad news about this. Genetically modified organisms are here to remain. We will keep them, we need them. We just have to be careful with what we do. But this is the same. If you have a knife, you can either put butter on your bread or kill someone. So it depends on us what we do with it. But gene transfer is a fundamentally important aspect of, let's say, supramolecular chemistry. I mean, let's say it's more medical, of course. And I think we absolutely need it. And I think all the young students who listen to me have to propagate this idea that we have to control these things, and we know how to do it, and we will use it in the future. Then let me say a word about supramolecular materials and biomaterials, because this would be quite a very interesting application for you. It happens that we have developed over the years what we may call supramolecular polymers. We had published the first paper on that in 1990. 23 years later, in 2013, a small company, Xeltis, in the Netherlands, in Europe, has developed supramolecular polymers to make them biocompatible, and has developed polymers of supramolecular type for the development of cardiovascular implants to reconstruct the heart of children who had a severe cardiac malformation. This was done. These materials were developed and were indeed inserted into children. And let me show you the picture of the first child who received this implant of supramolecular polymers. Here it is. This is Dominica. At this time, she was 4 years old in 2013, so now 8 years ago. And it was done by Professor Leo Bokeria. He did the implantation on October 23rd, 2013 at the Bakulev Scientific Center for Cardiovascular Surgery in Moscow. Now, she looks fine. This is 3 months after implantation, and he looks very happy too. I can understand that very well. And we were also extremely happy to see that. Of course, not immediately, but the idea of supramolecular polymers was fruitful, especially also for the development of these possibilities of cardiovascular implants for children, thanks to the efforts of many people and especially of this company. So many children have been now implanted. This is more than 10. And the same company also made then heart valves, which have been implanted into children in Budapest, in Krakow, and Kuala Lumpur. So this is considered as a breakthrough in surgical practice. Let me also show you another property of supramolecular materials. Because the connections between the molecules are weak, you can break them, but then they can self-heal. They heal again. And here you see an example of a self-healing supramolecular polymer film. Here you have this transparent film here. You see very nicely transparent. You have the fingers of one of my Indian postdocs who was working on this problem when he was in my group. So somebody is cutting this film in two, and then you superimpose the two pieces, the two parts. You press here in the middle with your finger for a minute or so, and then it sticks again, and you can stretch it. So indeed, these are quite impressive aspects of self-healing materials. Now let's go back to the next step. We talked about molecular chemistry, supramolecular chemistry, but as I insisted at the beginning, the basic structure of a universe allows for something which is called self-organization. So on the basis of what we know from molecular chemistry, from supramolecular chemistry, can we now try to go into systems which would be able to self-organize, directed by molecular recognition, but what we have unlearned on that. So this is spontaneous but information-controlled way of generating architectures by specific recognition patterns. So it's a molecular recognition directed, spontaneous generation of complex architectures. An example which illustrates that very well is the formation of a virus. Of course, we have right now a virus which makes us a lot of problems, but the virus is not living. A virus is a dead object. A virus only living when it infects a cell and then uses the machinery of the cell to reproduce itself. But as such, a virus is dead. But it nevertheless has the features of being able to infect cells. Let me show you here the one example, and it was the first virus which was well known scientifically, the tobacco mosaic virus, which is built up from 2,130 protein subunits and one molecule of its RNA in the middle. These 2,130 protein subunits assembled spontaneously. The pieces are constructed in such a way, as you see on the right in more detail, that they fit side by side together. They bind together by molecular recognition between the surfaces of these building blocks, these protein subunits, and they can, as you see on the left, then assemble and form progressively a sort of a helix, which has a hole in the middle into which the genome of the virus is inserted. So this is happening spontaneously. We understand all the pieces of it. It's organic chemistry, it's physical chemistry, it is analytical chemistry to understand what's going on. And so we understand every piece in this thing. It is no magic. It looks like magic, but it's not magic. It's just based on the fact that the basic bricks, proteins, recognize each other and fit together in such a way that they make, like in this piece of pie here, they get together and then make a ring, a disc, and then a lock washer, and then they build up the final virus. This is a programmed chemical system. The formation of a virus from its building blocks is a program where the storage of information is on the surface of the building blocks. They bind together and they generate an output. So we can say that the program is molecular, it's stored in information of the components, and the operation is supramolecular. There's a processing via the recognition algorithm, the way in which they fit together, which is defined by the interaction pattern. Now, let's have a look at, just for illustration, how one can do that in a laboratory. And of course, many laboratories have been interested in trying to set up systems where you can have, where you can generate complicated architectures by spontaneously, but in a controlled way. And I take as example, which is a simple way of showing it, the fact that one can design molecules capable of binding metal ions, and the metal ions will be serving as connectors, as a cementing unit to bind these molecules together. So these ligand molecules interact with the metal ions if you design everything well to generate, by design, a specific architecture. Let me show you just some examples. For instance, one can make a double helix and an artificial double helix and an artificial triple helix from molecules, the brown and the blue. These are two molecules which wrap around each other. You don't see the midline which binds them. On the right-hand side, you have these three strands which are bound together by metal ions, which as you see in yellow here. You see these small pieces, these spheres in yellow. These are three strands held together. You can assemble, for instance, 9 silver entities, silver ions, using 6 ligands, and at each intersection there is an ion gluing them together, so that here you have 6 molecules which have 9 intersections, and the metal ions glue them together at these intersections. This is an interesting compound also. This is a sort of a very small cylinder, which is interesting because you can see that there is one type of molecule, the linear ones in reddish here. There's another type of molecules which are planar, the blue ones, and they are linked together by 12 metal ions, which sort of brings them together. And this is 19 components which go together spontaneously to generate the final architecture. All you have to do is to mix together the red molecules, the linear ones, the flat ones, and the metal ions, and spontaneously you will generate this cylinder here. You can have many others. It's just to give you some pictures which look nice and which have been done, and many, many more have been done, even much more complex ones than the ones I show you here. These were all made by us, but there have been many others made since then, and this is a field which has been extremely active. Now, this is also of interest for nanoscience and nanotechnology. Indeed, program self-organization can be implemented for the generation of architectures in a very well, and hopefully, in fact, the better you understand it, the better it goes, in spontaneous but controlled generation of very complex architectures. It is maybe not an alternative, but for sure a complement to, in nanoscience and nanotechnology, a complement to fabrication and manipulation at a nano level. Why? Because you can hope that one can set up systems like the ones I just showed you earlier, which are like these ones, which are very simple, comparatively simple, but which show that you can do it. You can design building blocks such a way that you generate a given entity at the end. And this is then, of course, the ultimate way of fabrication. You do not need, in principle, this works, you will not need anymore to make things, but you let things make themselves. Devices might be able, at least in large part, to fabricate themselves spontaneously. And this you can call the ultimate fabrication, is the self-fabrication. Now, here I spoke a lot about design. Design in chemistry relies on molecular information, programming in the correct way in order to generate functional molecular and supramolecular entities, and in a controlled, really, controlled is a very important word, the controlled, spontaneous, but controlled way of generating these functional entities. There's another approach which is one step further in complexity, one step further towards complex matter, which is that, what about doing it with selection? In other words, letting the system select from a diversity of building blocks, these building blocks which it needs to build itself up. In order to do so, and because binding it starts with a collision between objects, you have to have the best collision, the best fitting. In order to do that, that means in order to explore the diversity and the fitting, you need dynamics. Objects have to be able to fall apart and to reconstitute themselves in the same way or with some changes. So selection requires diversity and dynamics in the interaction. This then leads to a type of chemistry which is now developed for the last 15 years or so. It has been started in parallel by the group of Jeremy Sanders in Cambridge, UK, and by us in Strasbourg, which is chemistry where the objects the building blocks making up a given chemical object, making up the constitution of a chemical object, is dynamic. It can break up into parts and reconstitute the same or another one. And the fact that by the breaking up and reconstitution, it can generate something else in response to outside forces, I will come back to that briefly. This enables adaptation, and that is the way towards what we now are mostly doing, adaptive chemistry. And this is a next step, so to say, in the way towards complex matter. I will just say a few words about this adaptive chemistry before then concluding with some general remarks. This constitutional dynamic chemistry, the words are a bit complicated, but it exactly signifies what it should signify. Constitutional dynamic chemistry is the chemistry of objects whose constitution can break up into pieces and reform itself, or reform an entity which reassembles the components reversibly, but on doing so, it may change because there may be adaptation through variation in constitution in response to a physical stimulus or a chemical effector. These physical stimulus can be heat or pressure or something like that. The chemical effector can be adding some kind of a molecule to which would interact and change things. So because the objects are able to break apart, to reconstitute themselves or something somewhat different by incorporating something else, this makes it able to adapt to these outside agents. It has been used for developing new pathways for discovering biologically active substances, for making dynamic nanostructures, for making dynamic materials which can be adaptive, and technologies which can be adaptive. Adaptive materials, adaptive technologies. This is also something which is not well known yet. It's just starting, but making technologies which will adapt depending on what happens outside. This is, of course, something very exciting, and this would be something like properties of a plastic, of a polymer, going from liking water, hydrophilic, to not like hydrophobic, to change the relationship, interaction with water. This is something which could be really an adaptive technology. And these are three of the illustrations of the ways towards adaptive chemistry. So we have reached a point where we can sort of summarize this path from molecular matter, molecules, making molecules. This is, of course, what we have to do. We have to learn how to make molecules, to build up molecules. This is the basis of the craft of chemists. Then these molecules assemble, and then controlling the assembling of molecules into supramolecular entities followed by organizing, because this is then generally is an organized entity, depending on how you build it up. Dynamic, meaning that you have to keep dynamics so as to be able to search and to select, so as to allow for adaptation. And these are steps towards complex matter. So this is what I wanted to describe you for these steps. We are now mostly studying adaptive chemistry, but it is the next step is to, all the things I have shown you up to now deals with systems which are at thermodynamic equilibrium. Life is out of equilibrium, as you know. Life is a non-equilibrium system. It's operating far from equilibrium. So we have now to introduce also in the next step non-equilibrium systems, and a number of laboratories are studying that. We have also developed some approaches, but it's still a long way to go to generate off equilibrium, far from equilibrium systems. And this is the next step. The adaptivity being not, as I would say, passive, like what I have shown you up to now. This would be passive self-organization, where you generate thermodynamically stable objects, and then having active self-organization, which is out of equilibrium, far from equilibrium systems, like a living organism. Now, let me now come back a little bit on more general considerations. I suppose you have by now understood that the essence of chemistry is not only to discover what is around us, but also to create objects which do not yet exist. The book of chemistry should be written in addition to be read. Of course, we have to read the book of the chemistry developed in the universe by the evolution of the universe and by nature around us. But the chemists have the power to write the next chapters. Like when you talk about music, the score of chemistry has to be composed, not only to be played. Of course, you have to play them, what already exists, but you are able to compose it. So chemistry is a very creative science. It has a intrinsically creative power, which I can represent again by a sculpture of the famous French sculptor, Auguste Rodin, where you see here a hand of the artist expressing out of a stone a sculpture which is not contained in the stone. The stone has nothing inside. It's the hand of the artist which brings it out, or let's say, which fashions it, which makes it, which shapes this sculpture. Chemistry does the same. Chemistry uses the bricks of visible matter to build up its objects. So one can say that chemistry is the art of matter. Chemistry is like an art, it's creative. It makes objects which do not exist yet, which is the definition of art, and under the hand of the chemist who is able to generate these objects. Now, this kind of considerations, if we want to stretch it a bit, is already included, so underlined by a fantastic sentence written by somebody you certainly all of you know, Leonardo da Vinci, who had the famous painting La Gioconda in the Louvre Museum, which everybody wants to look at. In 2 years ago, in 2019, it was the 500th anniversary of the death of Leonardo da Vinci. He wrote a sentence which on the top is given in Italian, its original language. On the bottom in blue, it is translated. Let me read it slowly and see. Where nature finishes to produce its own species. These are the entities generating in the course of the evolution of the universe. But then comes something very important. Man begins using natural things, using the bricks of matter. We have now seen the elements, the periodic table of the elements of Mendeleev, in harmony with his very nature. You can interpret that the way you want, but it means for me, and I think it should mean, that in harmony with the laws of physics. Nature is built up with given laws. The structure of our universe is such that it has given laws, and these are the laws which control the way in which things build up. So it has to be obviously in agreement with these laws. And the ending then is extremely strong for somebody like Leonardo da Vinci, who was an artist, as you all know, but also a scientist and an engineer. He writes at the end to create an infinity of species. This is a very strong ending. Creating is something which is usually limited to art. No, science is creative. An infinity of species, meaning there are lots and lots and lots of things to discover which have not yet been made, but which can be made thanks to progress in science. In the Greek mythology, you have also a complicated mythology in India. I know that, it's very complicated. Here is Prometheus, one of the gods who runs away. He's sort of a traitor, so to say. He steals the fire of knowledge from the gods and gives it to mankind. Here he's running away, looking over his shoulder to see if the other guys are not going to run after him and trying to catch him. But he succeeds and brings the fire of knowledge to mankind. There he is, progress in science, holding high, showing high the fire of knowledge. Now, that's fine, but we cannot give it back. Knowledge is something which sticks with us. What we know, we know. We cannot give it back. No way. You cannot just erase knowledge unless you destroy the earth, of course. But there's some other planet somewhere in our huge universe where there will be people who have discovered that the universe is made of the elements of the periodic table of Mendeleev. You cannot give it back. You cannot just erase knowledge. You have to handle it and you have to learn to live with it. That is not simple. Mankind has misused knowledge, but mankind has also used knowledge in the way it should be used, in a fine way for the progress of mankind. But it has also been used for bad purposes, for war, for killing. And as I said earlier, for a knife, I would also say that evolution, and I like to say that has given us hands. Evolution has given us hands. With these hands, I can caress or strangle. Depends on what I want to do. I can have a nice gesture of caressing, or I can hit and strangle. So we have to decide about what we do with it. And this knowledge which science has brought to us, this quest of knowledge, is leading us to the control of our own destiny. What I mean also by that is that now mankind can do what spontaneous evolution did before. This is the you might call it post-Darwinian evolution of biological objects, where scientists, where mankind is able to control its own destiny, able to control what it is, what a human being is. And this is something which is certainly our future, but we have to learn how to do it. We have to be careful and we have to think about. We have the power, but we have to learn the use rather than the misuse. In the past, and you are a university, you are engaged in transmitting knowledge, educating the citizens. In the past, transmission of knowledge, so to say, went from this hand, the ape, to the Homo sapiens, to mankind. In the present times, from one human being to another human being, across generations. For those who don't know this is a painting by Michelangelo in the Sixtina Chapel in Rome. Very famous. Now the next one, what is in the future? What is there in the future for us? In the future, let me just show you this. I do not mean that in the future we will only shake hands with robots. Of course not. I just want to illustrate this by saying that we will build up artificial entities which can be companions, and we will modify ourselves, not necessarily by introducing metals into a human body, but by changing the molecules, the molecular structure of the body. Furthermore, I can already tell you, and you realize that very easily, some of us may have implants of teeth. Some of us may have another lens, an artificial lens in their eyes, made of a polymer. These were modifications. This is not us anymore. This is a piece of metal. This is a piece of polymer. And if you push it and say you have somebody who has a heart implant, if you have a heart implant from a donor, you cannot tell your wife, I am giving you my heart, because it's not yours. This is just a pump which has been installed. And I'm sure in the future, we will be able, by the progress of developmental biology, for instance, to make artificial hearts, biologic way. Looks like science fiction, but I'm convinced it is not science fiction. Now, let me finish with a mathematician. I didn't say much about mathematics, but there is a very nice sentence I wanted to bring to you by David Hilbert, a very famous mathematician. He is buried in Göttingen in Germany, and here is his tombstone and in Göttingen. And on the tombstone are written two sentences here. What do they say, these sentences? The top and the bottom. The top, wir müssen wissen. We must know. That's really what we are going for. As scientists, as researchers, we must know. This is what drives us. But what is even stronger is the second part. Wir werden wissen. We will know. This is the intimate conviction that we will know. We will progress. The beginning is we must know. Sure, we must know. But then you have to have the drive that we will be knowing. We will conquer more and more knowledge, hopefully for the good. Another aspect, and the final aspect of science, I would like to stress for the students especially, that science has no borders. Science is a human activity which has a fantastic property, feature, that it is everywhere the same. A scientific step is valid everywhere, not only on this planet, but on other planets, in the hands of other entities, of other living entities, because the laws of nature, the laws of the universe are such that something done here can be reproduced somewhere else. And also, there are no borders for scientists. We are a family, a worldwide family. The students should realize that I think as people who are already advanced in their career, we know that because we're going around the world. but for students also, you have friends, people everywhere you can be friends with. So science brings us knowledge. We will know. I will encourage students to participate because science shapes the future of humanity. And science is across the whole world. This small, our home here, this small planet in a little piece of the universe, in one spot in space and in time. Nevertheless, science has been developed here, and in other places in the universe, science will be the same. Developed differently, probably, but the science is the science. And science has no borders. Science is for everybody. Science is universal for all of us. Thank you very much.
Thank you, sir. This insightful lecture was not only captivating, but also extremely informative and thought-provoking for budding scientific minds. I can sense that the audience was well dipped in the sea of knowledge you shared with us. Sir, with your permission, can we open the session for discussion?
Yes, sure. Yeah.
Okay,
but
sir.
I think the best is probably that somebody picks up. Okay, let's continue. But somebody picks up the question and repeats it because sometimes it's been. Okay, let's start and we'll see how it goes.
Okay, fine, sir. So, sir, we have received many queries in the chat box, but before going to those queries, I want to take the advantage of being host of the session. So, I would like to ask, sir, one question from you. Getting the Nobel Prize is the biggest dream of every researcher. So, can you please guide us how one can prepare himself to work in that direction, sir?
I think number one should not do science for that goal. It comes or does not come. You may bring your contribution to science, that's important. There are many other people, scientists, who should have gotten the Nobel Prize. But as there is only one chemistry prize, maybe with 3 people, but not more per year. Of course, there are many more other scientists who have done fantastic work and who deserve the prize. If you get it, of course, it makes you very happy because it usually means, by the way it functions, by the nominations, that the other scientists have considered that what you have been doing is important for science. And that's the way it operates. You cannot, of course, you know that, you cannot be a candidate for the Nobel Prize. There are no candidates. You have to be nominated by many, many people. It can take a long time. Sometimes it takes very long time. Sometimes it's very brief. A very long time has been taken by some, for instance, the electronic microscope took a long time. Barbara McClinton, she was the lady who studied gene hopping. This was also a very long time. And then sometimes very short, like for instance, high temperature superconductors in a few years got the Nobel Prize. It's just a question of, maybe sometimes scientists propose something and this world of science is not ready for it yet, cannot see the importance. In other cases, the importance is seen immediately. And then, of course, the prize comes quickly. But the 2 prizes, the merits are the same. So I would say, what is the recipe? There is no recipe. One is easy, work hard. You have to work very hard. You have to be passionate for what you're doing. You have to be able to resist when it doesn't work out, to resist disappointments. We all scientists, we know that we have many disappointments. Things just, the idea was no good. Or the idea was good, or we didn't try it the right way. So all this come together, and the main thing is work hard. But now wait a minute. When this question is asked, often by students also, I give them 3 advices. Number 1, don't miss the train. What do I mean by that? I mean that when you do your science, There are, of course, you work towards some idea, something. But sometimes there's hidden behind what you want to do, something more important than what you were thinking of at the beginning. So when the train passes, notice that the train is there. Don't miss it. This happened to me several times, that something was behind what we did and which was more important finally than what we wanted to do. We must have missed many of them. We don't know it, of course, but some we did not miss. Number one. Number two, do not jump on the train which has already left. Many cases, science is progressing, and it happens that it is an easy way to just jump on a train where there are lots of people. Because, there's lots of people, that means it's important, sure. And I think one should do it to some extent. But also, originality is not to jump on a train which is already full and has left. The third thing is think perpendicular, orthogonal, this way. That means question what you know, questions what is being taught to you, and try to do the opposite or try to do a perpendicular thinking. Usually it is stupid, the outcome, because science is well-based and so on. But sometimes it opens to you new doors, just thinking differently, thinking, as people like to say, out of the box. I call it thinking perpendicular or orthogonal, trying to put into question what you know and test the idea that thinking the opposite may be very fruitful. So these are my 3 comments.
Right, sir. Sir, one chemistry faculty, she is asking one question, that nature is able to perform number of diverse catalytic reactions in presence of enzymes. How do you foresee supramolecular catalysts could replace enzymes in many commercial biochemical processes?
That's a very good question. That's by somebody who knows supramolecular chemistry. There's somebody, an advanced chemist already, who asked this question. Yeah, I think, let's say, the way we function, an organism functions right now has sort of been optimized. Evolution has more or less optimized the reactions enzymes are doing, because it's not only the type of reaction, but it's the rate of the reaction. And it's also the localization of the enzymes, which are important. So it is, in principle, not easy to replace a natural enzyme by a supramolecular, by an artificial one. But there is also no reason to doubt that it can be done. It is not easy because, as you know, when you introduce a new chemical entity into a human body, there's a lot of controls you have to follow. It has to be sure that it is not toxic, that it has no side effects. But in principle, it's doable. I can see as a first step something which has already been done by some people many years ago, but in fact, in some respects, I think that it has perhaps not been pursued enough to take a natural, a normal enzyme and modify the active site a little so that it does something else. For instance, replacing an OH by an SH, or replacing one basic amine group by another group, an imidazole by some other one, some other kind of oxazole, whatever, another group. So one can see in the modification of enzymes of the active site a way to introduce other functions. But of course, there are problems. One of the problems is that these compounds will not stay around a long time. So they will be destroyed by the stomach, for instance. This is why the approach which might work, and which in fact is used already in animals, that is doing genetically modified organisms. There you introduce a new biological property, a new enzymatic property to synthesize other type of, for instance, to make a goat produce another protein. This has been done. And so that is probably the way to do it, rather than making a chemical, a supramolecular enzyme. The way to do it, first, if you want to change enzymatic features in an organism, is to do it through gene transfer. That is the most immediate way of seeing it. It can also be done otherwise, for sure, but it is certainly much more complicated because of the hurdles, the restrictions from the moment you go into a human body.
Right, sir. Sir, with a personality like you explaining the queries, the flow of question is not going to stop. But due to shortage of time, we will not be taking more questions now. So once again, thank you, sir, for this wonderful talk and question answer session.
May I make a small comment? Just something so that, of course, for you it's evident, but for the people who are not advanced in chemistry, the difference between molecules and supramolecular entities. I usually say the following. A molecule of water is something very simple, H-O-H, okay?
Right.
A single molecule of water cannot freeze, cannot boil. It's a single molecule. But if you have a glass of water, the glass of water can freeze, it can boil, it has a viscosity, it has a refractive index. Why? It's still water. Yeah, but it is now water where all the molecules interact. So from one level of complexity, the isolated molecule made of 3 atoms, you go to a totally different level, which is deduced from the one below. Putting molecules of water together makes them interact and gives them features. The glass of water has features which the molecule of water does not have. And this goes, of course, it's just a beginning, sort of very simple beginning, but one can then sort of in one's mind extrapolate and say, okay, if that is possible, the next step of complexity and the next and the next and the next and the next will end up by something like thinking, which is, of course, the highest function we have. What is important for us is what we have between our 2 ears, the brain. The rest is pumps and pipes and things like that, you know. I'm sorry to say, of course, the pumps are important. The skin is important. People could be nice or not so nice. So there are lots of things which in the envelope, but nevertheless, the important thing is between our 2 ears.
Absolutely.
Okay.
Sir. Thank you, sir. Now, I would like to invite Honorable Pro Vice Chancellor, Professor Dr. B.S. Sohi, to present the vote of thanks.
Hey, Professor, it's of course an honor to offer a formal vote of thanks to Hon'ble Nobel Laureate JeanMarie Lehn, and it's very rare to be face to face with such personalities of the world. I appreciate one point in you that is should be a guiding line for everybody is you are yourself discovering yourself, I should say. You experimented with music, philosophy, then learning 4 or 5 languages, then ultimately with little experience of experiments of chemistry at home, you thought that you are meant for or you are made for chemistry, and that's how you landed into and succeeded to the pinnacle of this. That is one lesson we should learn, and the parents must learn that we should allow the people to learn or discover themselves and then decide what they should be in life. That is one very, very important lesson we have. We started with the universe saying 95% dark matter and 5% visible matter, and this 5% visible is enormous to be comprehended by us, of course. Rightly, you created the link between physics and biology as a chemistry, and that is what normally people don't take it. Taking chemistry in isolation is something not appreciated. So taking from the organization of the universe and this organization of this through the electromagnetic these forces, then you came back to evolution of the chemistry, that is how the matter, the molecules, of course the atoms and all those, the science of structure and transformation you have talked about. Then, of course, Mendeleev's chart is a milestone in the science. Then you talk about molecular storage and in the science of informed matter, that is the chemistry. And then you emphasized about choosing between the basic science and applied science. Wherever you are working, applied science comes from basic, but wonderful things happening, drug discovery, medical diagnostics. Then gene transfer is one, the gene therapy. I don't know whether genetically modified organizations will be good or bad for the world. It are, as you said, they must be taken as it is, but it is how the world uses it. Then biomaterials, cardiovascular implants, self-healing materials, self-organizing materials, programmed chemical systems. And then you talk about nanomaterials, nanoscience, nanotechnology. That's from fabrication to prefabrication, and that is how the life is changing. Then, of course, the chemistry by design information, and then by selection, all those that will lead to adaptive chemistry. I can't really comprehend everything because I am not a man of science. So, but anyway, this is what I could comprehend, that things are changing very fast. Science is going at a very fast pace, and that's how it drives the world. You have talked about the future of search for the biological active substances and self-organization of the dynamic nanostructures, all this. And you ultimately said that chemistry is an art of matter, and it's up to the chemist how the chemist gives a shape to it. And of course, then you said that knowledge always sticks to us. That is always there. Unless you gain it, you don't have anything. So use of knowledge by the mankind is all important. It is a lesson to the youngsters that they should acquire knowledge. They should be aware of what is happening, going by the trend, as you said. All this is that's a matter that you have tried to put across in a very simplistic way and very systematic way, which even people like engineers like me could comprehend quite a bit, and can appreciate the role of chemistry, frankly, appreciate the role of chemistry in everything. So that's a wonderful thing. Then you again told us that how this evolution went on from molecule to supramoleculars, organized, dynamic, and adaptive matter, and all that. And then how will the future science research will go towards the complex matter. And a lot of things are there to be explored. And who works, it's all there, and who gains out of this, it's all there. So that is a great thing. People like you can only comprehend, put the things in a comprehension that we can understand. Sir, on behalf of Chandigarh University, on behalf of our Honorable Chancellor, Honorable Pro-Chancellor, all the faculty members and students, we are grateful to you that you have spared that much of time, which normally people don't have it or don't really care to spare. But this is for the benefit of the youngsters.
So thank you all for sparing your valuable time, and now the session is over.